to mute your mic. And Dr. Anamika has joined. Please mute your mic. Please mute your mic. Okay, okay. You can start now.
All right. Can you please mute your mic? Yes. Today we have a dear friend, a Bhatnagar Awardee, Dr. PS Raghaya, with us. We are proud of his achievement. He worked in IMTech in the 1980s, right from 86, 87. He was working in IMTech, and I also joined him in 1987, October. So we are very good friends since then. I'm really happy to welcome Dr. Raghaya today. Now over to Pradeep Kaur, who is the coordinator of MBSI Chandigarh. Pradeep, please start. Dr. Pradeep?
Yeah, she has left. I don't know. But she will join again. 1 minute.
Just
1 minute.
in 1 minute, yes.
Because she is not there.
Good morning,
Dr. Pradeep.
She was there, but something has happened.
She'll come.
Okay, okay. In the meantime, I am very happy. Actually, I should say thanks to Swaranjit Singh instead of Raghavan sir. The reason is that a big personality like Raghavan is on our platform only because of Swaranjit Singh. His communications, his relations are very helpful to microbiology society. And of course, Raghavan is a Bhatnagar Awardee. And if such person is giving knowledge to our common microbiologist and biotechnologist, we will enjoy it. And it's a great asset, today's lecture. Again, we are circulating it on YouTube. And I'm sure many more students will take advantage of your lectures.
Gross profit income 1,62,356 hours.
1 minute, I will. Yes. So, Dr. Pradeep Kaur is again not to join, sir. So
She is having some internet problem.
okay,
So,
okay.
maybe we can start with the talk,
Correct.
and she'll combine the welcome and vote of thanks together. No problem.
Yes, yes. Okay, thank you.
So I welcome Dr. Raghaya, a Bhatnagar Awardee, and a friend, Dr. PS Raghaya, to start his talk. Thank you.
Dr. Raghav is a co-host, so he can present his PPT. Thank you.
Your mic is mute. Raghaya, your mic.
Yes, yes. Thank you, Swaranjit, for inviting me for this MSI. So I'm grateful. And we remember that our assistant from a long time. And this is the time where I can interact with the students actually. Okay. So I will just share my talk. Okay.
Yes.
Just hold on. Is it visible? My slides are visible. Can anybody confirm?
Yeah, yes, sir. Yes, sir.
Okay, fine. So as you can see, today's topic is computer resources for managing drug resistance. So I'm not expert in the biology or microbiology, but my major challenge is towards the developing the computational resources. How to solve the biological problem. Swaranjit Singh know very well. I joined the IMTech Chandigarh as a computer scientist to provide a service to the institute. So I was the head of the computer center in IMTech Chandigarh, and where I slowly learned a little bit about biology and try to contribute some software for the community. What I realized actually that in India, we have the lack of confidence or insecurity. So we never share our resources with the others. We don't want that there should be competition. But in this process, we lose a lot of information in real life. So when I came into the research, it was by hobby. It was not for any career or any type of thing. So what I did actually, whatever softwares I have developed during my research, I make them free to the public. So all the resources are freely available to the community. Please mute the mics actually. Okay. So today I will talk about the computer resources for managing drug resistance. So I have divided my talk in 4 parts. First, I will talk about the concept of drug resistance. I think most of you are biologists, so you know the concept of drug resistance. I will not go into the detail, but what I will talk by using the computational resources, how we can predict, how we can identify whether a particular strain is resistant or not, or which gene is responsible for the resistance. Because I'm looking from everything from the computational point of view. Okay. So first I will talk about the concept of drug resistance. Then I will talk about the 3 alternate techniques. Because here when I talk the drug resistance, I'm talking about drug resistance against the antibiotics or small molecule based drugs. So I am talking about 3 types of therapies beyond the antibiotics or small molecule based drugs. These are peptide or protein based therapy, vaccine and immunotherapy, phage based therapy. These are the 3 therapies. Please remember one thing. I am not the expert of all these fields. I am learning these fields so that I can contribute more and more databases, software in these areas. So first, this is a well-known slide. You all know that why drug resistance is there. Sometimes it is because the membrane become the permeability change so that antibiotics or small molecules cannot enter inside the cells. So that drug resistance is due to the membrane permeability change. Another thing that even if you enter, the bacteria release certain enzymes like beta-lactamase is just one enzyme which degrade the antibiotics or small molecules so that drug is not effective. Third thing is that drug, the efflux pump released, change the drugs so that what happen when there is a drug molecules, then the efflux pump, they secrete it out so that it cannot stay for long in the inside the cell actually. So that's another reason for drug resistance. Another reason is the target is modified. Let's say let's say drug is binding, have the target where the protein and DNA is binding. If it is changed, then drug will not be effective. So these are some major reasons why there is a drug resistance. So I'm not going into the concept of drug resistance. Question is, what are the computational resources which you can utilize for your work day to day? So these resources broadly can be divided in three categories. One is databases. There are number of databases are available where the gene, DNA, mobile, genetic elements are available of drug-resistant strains of the bacteria as well as the sensitive bacteria. Protein and nucleotide sequences, beta-lactamase, all the databases, these maintain this type of information. Another is basically, let's say nowadays, due to the metagenomics or genomics, you can sequence any bacteria or even the group of bacteria in one go. You heard about the next generation sequencing, where we can take the whole set of the culture and then we can sequence it. Okay. So the question is, can we predict whether the strains if of a particular bacteria or maybe fungal is resistant or not? Okay. And third is a pipeline which provides the complete information. So if you are really interested, then these are the major databases we have compiled from the literature. These are not developed by us, developed by some other groups basically. Okay. Argo is a database of antibiotic resistant genes. ARDB is the information about antibiotic resistant genes. So these are the different databases. You want to know which genes are involved in the resistance or not. These databases provide the complete information. So you can get the information. This is important for you, even if you are not bioinformatics, doesn't matter. So you should know at least from where, as a microbiologist, you can get the information. That's very important for you actually. In today's time, if you work without the knowledge of the existing resources, then you are not able to utilize your full capacity. So it's important for you to learn at least what are the resources, where they are available, so that you can utilize in your science. Same way, there is a number of softwares are there where they predict whether a given gene is drug resistant or not. Okay, because there is a continuous mutations are going on. So whether the mutated gene is drug resistant or not. So there are a number of softwares. I have listed some of them here actually. These slides will be available to you. So if you are interested, you will get that information later on. As I told you in the beginning, one of my major contribution in science is that I try my best to make everything freely available to the public. So whatever the resources I have developed, even lectures, I make everything free to the public so that more and more people can learn it. So our new generations can learn from the old generations, basically. That's one of the important part in the science, actually, which we are missing sometimes, that our goal is not to make our own careers, but our goal is to train our coming generations so they can learn it and they can learn fast, actually. So all these slides, all resources, whatever I have compiled are freely available. You can use them, actually. So these are some examples. So anyway, if you want to follow it systematically about the drug resistance, then this is a review we write 2 years back. That's in the drug discovery today. That's on the computer resources in the management of antibiotic resistance. So this paper is available. If you need the reprint, I can provide you. That's not a problem. Okay. And you read in the detail, you will get the complete comprehensive information about the drug resistance. Fine. Now we know what is the problem. You identify the problem. That's fine. But question is, what is the solution? So there are a number of alternate ways of, there is a number of alternatives for the antibiotics. So I am discussing 3 possible options. One is basically protein peptide based therapy. So nowadays, this therapy is basically increasing with the time. So first I will talk about this, then vaccine, and then I will talk about fast based therapy. So it is, let's say, I will say when I start in this area, I start maybe 7-8 years back. When I talk to some chemist, they say, no, there is nothing is there. where the protein can be used as a drug discovery. Why they say give this statement? Because they have the limited knowledge. Sometimes please remember, if you are coming into the science, listen to everybody, there is no harm actually. But don't take the knowledge as it is. Sometimes it may be confusing. Best friend, the best way to learn in life is literature. Go to the literature, read the recent paper, you will know what is the recent trend. Because I talk to the chemists which have the old time trained people, they have a hardly knowledge about the protein-based therapeutics. So anyway, I read the literature and I realize nowadays a lot of molecules have been approved by FDA which are based on the protein or peptide based actually. So what we did in our group, we collect and compile this information from the literature or FDA approved sites, and we create this site. This is called THPDB actually. Okay, so this is available from this site, and believe me, it was published in 2017. By this time, we have around 340 citations. So now you can understand how it is used by the community. So it is heavily used by the community for their work. So somebody want to understand what are the proteins which have been approved for the therapeutics and all sort of things actually. So it is important to compile these informations, and this information is available in the form of the database. So you can browse them, you can search them, all the things. Now question is for you, you want to know that what is the application, how protein or peptide are used in real life as a therapeutics. So broadly, I'm saying that broadly, you can, it is defined in the 4 groups. Group 1, maybe in natural form, the particular protein is defective or particular protein has some problem, we can provide that protein from outside. Protein from outside. Example is insulin. If somebody is not able to produce the insulin, or our beta cells are not there or not able to produce, then we can provide the insulin from outside. That's just one example. Or maybe some enzyme or regulatory activity. So this is one application where the protein plays an excellent role, and number of proteins have been approved for the therapeutic use. Okay. Another area is drug delivery. Let's say just now I talked to you about the there is a drug resistance due to the membrane of the cell. So if you provide the cell-penetrating peptide and attach the drug, the cell-penetrating peptide have the property, they can break even the resistant membrane, and they will enter into the cell with the drug, so that drug delivery can be done actually. So here is an example where we can use the protein or self-assembled peptides for delivering the drugs. Third application, everybody knows about the vaccines. We know about the vaccines, and you know about the COVID-19, everybody knows it. We have been saved due to the vaccines, not due to the drugs basically. Okay, because we got the Covaxin, Covishield, number of vaccines have been given to us, and then we got the protections basically. So this is another area. And fourth area, I'm talking about major areas, is diagnostic agents. So sometime in certain disease, peptides and proteins are found in the abundant range, means more in the quantity or less in the quantity. And based on that, we can say the particular person has the particular disease. So that is called disease diagnostics. So protein and peptide concentration can be used to measure the particular disease basically. So these are the basic things. And I want to make the statement here that chances of failure of protein or peptide based drugs, therapeutics, is much lower than the small molecule based. So nowadays, this trend is changing. rather than small molecule based, we are making the protein or peptide based therapeutics, which is approved by FDA. So this is just an example. Our group is contributing from last number of years in different areas actually. It's not, our group is a little bit different than the most of the research groups. So here is a message actually. I get a lot of criticism for the number of top scientists because I do not follow any rule regulations. According to the well-known hypothesis, a scientist should focus in a particular area and the person should go into the deep and deep. I do not follow this story because
இந்த வாழ்க்கையில சாதிச்சவங்களுக்கு டாக்டரேட் கொடுப்பாங்கல்ல, அந்த மாதிரி எனக்கு நிறைய சாதிச்சுட்டு அப்படியே யூஜி கொடுத்துட்டாங்கக்கா.
Please keep the mic mute.
வந்துட்டான் பேர சொப்பையன்.
Please keep the mic mute.
Bro.
Okay. So here it is important that we believe in ourselves actually. I will tell you, number of people criticize me that I work in this area, that area. So we have to understand, I love to work in the different area because I work for knowledge. I enjoy to read more and more fields. So that's why I go into the different fields and then I contribute it. So you have to decide yourself what you like in your life, on what is your interest actually. That's very important actually. Otherwise, after some time, you will be tired actually by doing the science, because most of the theories in the real life, they are based on their own experience. But individual is different. So you have to know about yourself first actually, what you want and what is your interest actually. Based on that, you have to do the research. So we contribute in the different fields. So here is a self-penetrating, designing self-penetrating peptide. That's fine. Experimentally validated peptide database is there. But if you want to design new self-penetrating peptide which have the desired property, then we develop the tool, the cell PPT, which allow you, which says by mutating this particular amino acid, then cell penetration will increase or decrease actually. Okay. Similarly, number of tools have been developed. This is one of the recent tools from our group, predicting the anti-cancer peptides. Let's say, like you normally have the anti-cancer small molecules. Okay, here I'm talking about the anti-cancer peptides basically. So this tool was earlier developed, old version in NTCP, but there was a lot of demand from the public and a lot of citation was there. So we updated it, and this new version is called NTCP2, is available from our group. Okay, so if you are interested, you can use it. This site provides the prediction, design. Design means, let's say you have a peptide, you want to make the mutation so that it can become the better anti-cancer peptide. Protein scan, you know that particular protein have the property of anti-cancer property. Now you want to know which region of the protein is responsible for anti-cancer activity, so that you can selectively select that region which is an anti-cancer peptides basically. All right. Then motif scan, some motifs are responsible for the anti-cancer activities, so you can select it. Similarly, you can go for the antifungal peptides. So these are the simple tools where you can simply click on the predict, put your amino acid sequence, it will predict whether it's an antifungal peptide or not. So it's very easy. Sometimes you may feel that I am biologist, microbiologist, I have no knowledge of the computer. How can I use it? We design this tool user-friendly. Even if you have zero knowledge of the computer, just you know how to do the browsing, you can use these tools actually. And what is the advantage? Sometimes you don't know, let's say you have a big protein of 200 amino acid or 300 amino acid. You don't know which region of this protein have the antifungal property. Then submit this sequence to our server. Our server will predict these are the possible regions where it has the antifungal property. Then synthesize only the regions, predicted regions, and try experimentally. Please remember one thing. Prediction doesn't give you the guarantee that it will be antifungal activity 100%. But it gives the guarantee that chances is high that it will be antifungal because it has been predicted by software based on certain properties. So it's important when you are doing the science, experimental science, you have to use these techniques. Same way there is a other like antibacterial peptide prediction. So we have the huge number of series of softwares, web servers. So you just visit my site, you will get them. Even if you put on the internet the particular activity, our site will come up, you can use them actually. If you can't remember, it's fine, no problem. One of the challenge, like when you talk about drug discovery, same way in peptide protein-based therapy, the challenge is half-life of the peptides. This is the biggest problem with the protein or peptide-based therapeutics. Why? Because in our body, we have the large number of enzymes which degrade protein and peptides easily. So we have to select the peptides and proteins which have the long half-life. Because if it doesn't have the half-life or limited half-life, then what will happen? It will be degraded fast. So it will not be effective. So it is important to predict the half-life of peptides. Sometimes, in order to increase the half-life, we do the post-translation modifications, or maybe let's say N-terminal modifications, C-terminal modifications. But major question is, what will be the half-life? So this server, PepLife, will help you to predict the half-life of peptide after making certain mutations, or it will suggest what type of mutations you have to do in your peptide so that it have the longer half-life. Another problem, though we claim that peptide and protein have the less toxicity in comparison to the small molecules. That's true, because they are natural molecule and they will degrade after some time. But they have no toxicity. That's not true. Because this is a sometimes misconception we have always, that if it is a natural product, or if it is a natural, let's say, biological system, come from protein from biological system or any system, it will not toxic. That's wrong. It may have the toxicity. So what this server provides, that it help you to identify peptides which are toxic in nature actually. So it's important because let's say if you have designed some peptide which you feel is a basically best peptide for therapeutics and you want to take it to the clinical trials. If it is toxic, then it will fail in the clinical trials. And we spend a lot of money in the clinical trials. So before going to the clinical trial, it is better to predict the toxicity of these peptides. And if possible, mutate the peptide in such a way that toxicity of the peptide is reduced or removed. So this server will help you to identify the or design the toxic-free peptides basically. Sometimes we feel that toxicity means only cytotoxicity, but there are different types of toxicity out there. One is basically hemolytic peptides. Maybe your peptides are not toxic to the general cells, but maybe they are toxic to the RBC. So this server will allow you to predict the hemolytic potency of your peptides. That is called HemoPI. So you can use this server whether it will be toxic to the RBC or not. Similarly, because I cannot cover all the tools developed by our group, there's too many actually. Another way is allergy. Allergy is another part of it. So we have the tool called LGPRED2, which can be used to identify the allergy of the protein molecule. So anyway, it's not practically possible to cover everything. So if you have the time and you are interested in this area, then you can review this. You can read this review from our group, where we have tell about the in silico tools and databases for designing peptide-based vaccine and drugs, basically. So if you are interested, you can read this review. After talking to the protein and peptide-based therapy, the another therapy which is important is vaccine and immunotherapy. So what is the difference between vaccine and immunotherapy actually? Vaccine is a basically long-term plan where you generate the memory in the long term and provide the protection for the long term. In immunotherapy, in certain disease, it's changing too fast. So then immunotherapy is used for that purpose, basically. All right. So when we talk about the vaccine, then everybody know, because most of you are biologists, you understand the concept. So I'm not going into the detail. You know what is a vaccine. We take the whole organism, we kill it or annutate it so that it cannot propagate or it cannot replicate and give to the body. And body thought that particular bacteria or virus is there, and then they generate the memory B cells, memory T cells, and corresponding antibodies. So next time when you are infected in real life, then you have the memory cells, B cells, T cells, as well as the antibodies. They will fight against it. So the best example for this part for the COVID-19 is Covishield. I asked this question with the number of students in my interview that what is the principle of Covishield or Covaxins? Then they have no idea. So the Covaxin is based on the whole organism principle. What they have done, Bharat Biotech, they have taken the coronavirus, they annotate it so that it cannot replicate, and then they give to the body. So that's basically whole organism based vaccines. Sometimes you don't require the whole organism, you need the only one antigen of the organism. Then you can design the vaccine, and this is called the recombinant vaccines, which is based on the purified antigen. And good example is Covishield. I hope you know about the Covishield. Covishield is based on the surface protein of coronavirus. Surface protein of coronavirus. So that rather than using the whole organism, we are using only the surface protein of the coronavirus. Okay, so that's the second principle, whole organism, antigen. And third way is why do you design the vaccine even taking the whole antigen, let's say surface protein itself is a big protein of 3000 amino acids. Why you need the 3000 amino acids? You can identify the B cell and T cell epitope which have the length of 9 amino acids. That can also serve as a vaccine, which will have the minimum toxicity and maximum efficiency. So our group is mainly focused on the epitope-based vaccines. Number of vaccines have been designed against the COVID-19, though they have been not been popular in India, but they have been approved in other countries actually. Okay, so these are the principles actually. So when you talk about the vaccine, please remember one thing: you have to understand the immune system. So what is the immune system? When there is an attack from any microbial pathogen attack, it may be disease-causing agent, bacteria, fungus, parasite, or viruses. What will be happen? There will be two types of defense: non-specific defense and specific defense. In case of non-specific defense, there is a toll-like receptors or PRRPs, pathogen recognizing receptors, which recognize them, and then they provide the first line of defense as well as the alarming systems. Then there is a specific defense. In specific defense, it may be humoral immunity, which activates the B cells and antibodies. There may be cell-mediated immunity. This may activate cytotoxic T cells and T helper cells. Okay, so this is the overall concept of vaccine development. So here we have developed large number of tools. If you are interested to use them, you can use them. Let's say, for example, for T-cell prediction, we have the number of tools. These are the name of the tools and the brief description how they can be utilized actually. Then these are the B-cell related tool, and these are the vaccine agents. Vaccine agents is nothing. It's basically activate the innate immune system. That's called first line defense. So that activate our adaptive immune system. So that's basically vaccine agents. You will surprise in number of cases whether the drug or vaccine work or not, but the vaccine agents or adjuvants actually, drug adjuvants, they play the excellent role actually. So it is important to design better vaccine agents basically. And here, what we are talking about the peptide-based vaccine adjuvants or DNA-based vaccine. We have developed a number of tools you can utilize for your research. So the question comes, when there is a certain problem, what is our role? Are we working passively or are we working actively? Passively in the sense we are doing our job. It doesn't matter whether coronavirus is there or not there, or any other virus is there or not there. So why we should to bother? We should to do our research. No, our group is active research, which sense what is the problem society is facing. And then we try to our best to help the society. Whether we are getting the credit or not getting, it's not important. Important is whether we can help the society or not. So when coronavirus comes, what we did actually, first job, that within few months, we developed a web-based platform on coronavirus, this is COVID-19, to maintain the predictive diagnostics. What should be the PCR sequences for the different strains? What is a possible drug candidates? What are the vaccine candidates? So this is a paper, but this paper is based on this website. And this website was just developed to provide the updated information, complete and comprehensive information. What are the different genomes of the different strains, diagnostics, RT-PCR and other sequences or primer sequences, immunotherapy or vaccines, drug design and useful links. So that society can be benefited from this. Number of times I got the questions from my student as well as from others. Who will give you the credit when you are making everything open? Because we never ask anybody to give the credit to us. We put the things openly on the website. Anybody can use it. No need to give us credit or not credit, doesn't matter. So my feeling is like that actually. Why we are doing science? We are doing the science so that society can be benefited. Maybe somebody will use these tools, will design the drugs and vaccines. Okay, the person is not giving credit, person will popular, doesn't matter. But at least our coming generations will be benefited from this work, right? So that's very important to understand that part. So this is, if you are interested in the vaccines and immunotherapeutics, then this is the review we have written long time back, 3 or 4 years back, that you can use it. It provides the complete detail about the vaccine or immunotherapeutics. After talking all these areas, now I will talk about the new field called phage-based therapy. As I told you in the beginning, I'm not a systematic researcher. Our group do based on the trend actually. So far, let's say I will tell you that this is vaccine and immunotherapy. We are working from last 22 years. In around 2000, we started our activity. Protein and peptide-based therapy, we started 10 years back. But this phage-based therapy just started 1 year back. So this is a new field we have started. Why we started this new field? Because recently there is a trend change actually. I hope you know that fact that whole world is round actually. So we reach from where we start. So phage-based therapy is the one of the good example, where the phage-based therapy started much earlier than the antibiotics, but it has been dropped due to the golden era of antibiotics. Why? Because antibiotics are cheaper, they are very effective, so that everybody go to the antibiotics. But over the years, it has been realized that most of these strains nowadays are drug resistant to antibiotics. They are antibiotic resistant. Then these antibiotics are slowly dying their own life. So in that case, the old therapy, which is phage-based therapy, started long, let's say more than 100 years back, is again come into the picture. Now the people are again focusing on this old technology. So question is again what we are doing. We are the computer scientists. What we are doing here actually. So I will tell you our role. So this is the history of the phage-based therapy. So in 1915, discovery of phage by Frederick Tward. Then it was characterized, the beginning of the phage therapy, they have used it, okay. But the main problem started here, discovery of the penicillin. When discovery of penicillin come into the picture, then the new versions come, and then this is called golden age of antibiotics, where the use of antibiotics increase. There is another major milestone in the science actually by the Sanger sequencing, where they started to do the sequencing of the whole genome basically. So first genome which was sequenced was the bacteriophage actually. The bacteriophage is nothing, it's a basically virus which kill the bacteria is called bacteriophage. Okay. So let's say phage word itself is a basically virus which lies or kill the pathogen is called phage actually. That's a basically philosophy. So after the first sequencing was done, the whole genome was done in was bacteriophage actually. So phage first used to treat the drug-resistant bacteria in 1983. First time they saw a use that antibiotics fail in a particular case. Then they used this phage-based therapy to treat the drug-resistant bacteria. So why it is important? Because it can handle the drug-resistant bacteria. So then in 19 onward, that increase of drug-resistant resurgence of phage-based therapy started. One of the reasons why it is becoming so popular, the phage-based therapy, is due to the genome sequencing. As you know, that in this whole universe, there are more virus population than anything else. That's why number of bacteria, fungus, they are controlled by these viruses. If they will not control, then the growth of the fungus and bacteria or pathogens will be too high actually. Okay, and due to the sequencing, now we know the sequencing of the number of viruses. Some viruses are basically, let's say, they are for human health. Let's say COVID-19, that's not good for the human, but these are the viruses which kill our enemies, so they are good for our health basically. So number of new antibiotics decreasing because the drug resistance strains are coming. So most of the strains, the emerging strains of bacteria or fungus, they are basically resistant to the antibiotics, even for the new antibiotics actually. So increase use of the phage therapy is increasing. So first regulated clinical trial for phage therapy is started, okay, and already people are using the phage-based therapy for the product in this packaging and number of other use even in the animals. For human, there is a limited use so far, but in the future, it will be there actually. All right. After this, there are number of databases. If you are interested in this area, then these are the host phage instructions. Host here is a bacteria. Phage means virus basically. Instructions database. So this can help you to design the better host. The major challenge from the computer point of view here is to identify the right virus for the right strain of bacteria. That's a challenge. Because it's very selective. It's not that there are virus 10 to about 30 type of viruses are there actually, or maybe more. Not all virus kill all bacteria. There are certain virus they kill or lyse a specific strain of bacteria or fungus, basically pathogens. So it is important how to predict the correct or right phage virus for a given strain of bacteria, which is, let's say, for example, is drug resistant. So nowadays, due to the advancement in the sequencing technology, sequencing of bacteria is available, sequencing of virus is available. Challenge is how can we predict the interacting partners, right? So these are the basically major databases of phage associated proteins, because number of proteins are involved in the phage basically. So these are the databases. So I'm not going into the detail of each and every one. So these are the different tools. So if you are interested, you can come. Not too many people are working in this area, but I hope in next 10 years, a lot of people will jump to the new area actually. Because as a researcher, we always go to the area where there's a lot of demand is there actually. So nowadays, slowly the demand of antibiotics is decreasing, and we are looking for the alternative for the antibiotics. So this is a tool, we developed it, not still published. We have sent to the journal, and this software allowed to identify the right virus for the right bacteria. Means, let's say you have some drug resistant strain of bacteria, you want to find out the best phage therapy for that bacteria strain. Then our software will take the sequencing of two and will find out whether they will, this particular virus will infect the bacteria or not actually. Okay, and based on that, we can design. So this is a software. As I told you the beginning, whatever the work we are doing, we are developing the web server or software to the public so the public can use it. We are not doing science for ourselves. We are doing the science for the public, so the public can use it basically. That's all. So you can see, just because that's one of the problems we face most of the time, there is a certain type of complexity we have in our Indian researchers basically. Sometimes if you have the same tool or same database from India and from US, you will prefer to use the US one rather than using the Indian services, because we don't trust on each other. Okay. So, but fortunately, it takes time to us, but our tools are as popular, our softwares are as popular as any tools developed by anybody in the world actually. So why I'm showing this page, you can have a look that all the tools, whether it's a B cell apoptosis prediction, or it's a T cell apoptosis prediction, or it's a toxicity prediction, or it's allergy prediction, they are heavily used by the community and heavily cited by the community. So it is important to have the confidence in our Indian science actually. That's the most important part for our coming generations actually. We are not less than anybody in the world, only what we need to trust each other, to help each other, to work as a colleague rather than enemy of each other. Never consider each other as a competitor. Consider each other as a colleague or friends basically. That's all. Thank you very much. So I am open now for the questions. Yeah.
It was a wonderful talk, Raghav. We enjoyed every part of the talk. Dr. Raghav said you should like your work. Do work what you like, otherwise you'll be bored. We should help the society. society must be benefited by our work. This is a very important message by Dr. Raghav. Dr. Raghav, can I ask you one or two questions?
Most welcome. It's informal. Whatever the persons,
Right.
anybody want to ask, most welcome.
So in molecule-based therapy, can bacteria also be used? Bacteria?
Bacteria as a therapy, but
Yes.
what you will kill with the bacteria?
No, what I'm saying, in molecule-based therapy, can you use a fungi or a bacteria also?
Yeah, why not?
Okay. You talked about virus and bacteriophage.
No, bacteriophage is nothing. It's a basically virus which kills the
Yes.
bacteria.
That's right. No, what I'm saying, for this molecule therapy, can you use bacteria cell also?
Yeah, you can use it if you see carefully how the antibiotics come, which is
Yes.
released by fungus.
Right.
So they kill each other actually. Virus, fungus, or bacteria, they release the molecule which kill each other actually.
Yes.
So you can take the advantage of it actually.
Right. Because I read an article recently that even bacteria is used for therapy. For therapy. That's why I think bacteria can be used, fungi also can be used.
Right.
Okay, one more thing. Before clinical trials, before you do a clinical trial, we must be sure that the compound is not toxic. That's very important, right? Very important.
Right.
Can you mute your mic? Mute. Yeah, Raghav, what I'm saying, we have to make sure that the compound is not toxic before the clinical trials. Supposing there is some toxicity, how will it affect the program?
Now, the question is basically, in certain cases, even toxicity is there, we still give them. Let's say cancer drugs actually. Still,
Right.
they are toxic. But we know cancer is a bigger enemy than the toxicity actually.
Yes.
But
Yes.
in other cases, we will not give actually.
That's right. That's right. Now, Dr. Prakash, please ask question.
Good morning, sir. Sorry that I missed most of the part, but it was indeed a very interesting session. Actually, we have reported colistin resistance in lactic acid bacteria. And we want to go with the, we have in fact done the whole genome sequencing also. How Dr. Raghavan's lab can help us regarding this. So I just wanted to take his help perhaps.
Yeah, even you can go on the website, FastDB, and
Okay, sir.
here you can put your genome sequencing, and it will tell which virus, already known virus, can infect the bacteria and kill that bacteria. Or you
Okay.
want to design the antibacterial peptide, then
Yes.
surface is there that can help actually.
Wonderful. Yeah, I actually took the snapshots of your presentation also.
No, I will share the slides so that
Sure, sir.
to Swaranjit Singh, and he will share to the whole society.
Sure.
Yeah, actually, I was busy with this RC rehearsal presentation. I am from CFTRI, sir.
Okay, okay.
CFTRI, so yeah, thank you. Thank you, sir.
Any other question? Anybody wants to?
Good morning, sir. Yeah, I'm Tapti here from Kolkata. Swaraj sir and sir. So actually, I'm working on tuberculosis. There's non-tuberculous mycobacteria. So because of these MDR and XDR strains, sir, what computational part can we look to mitigate these problems?
No, let's say, for example, you are interested in designing anti-peptide, some therapy based on the peptide. Then we have the anti-TB pred actually, one software which can predict specifically for the tuberculosis, anti-tuberculosis peptides. Actually, you can design the anti-tuberculosis peptide and you can design the peptide-based therapy.
Thank you, sir. And what about phage therapy?
Yes, FastDB, if you have the sequence of that particular strain, then
Yes,
put
sir.
on our server, it will tell you which known virus can lyse this particular strain.
So we have 7 new strains identified through a DBT project, and they are put up on the server also. So like I can coordinate with you to find the resistance. I mean, drug resistance is tried. Now in silico is required.
Okay. You send the sequences, then we can help you.
Okay, sir. Thank you so much, sir.
We have
Good
time
afternoon, sir.
one more.
So
Thank you, sir.
Good afternoon, sir. I would like to ask regarding SVM threshold, how do we decide in most of the softwares I've seen for prediction, either the SVM threshold is default in 0.5 or it may be in 0. But how can we come to a decision of what threshold should we choose?
Okay. Normally, let's say how we decide that what should be the threshold. Let's say in our group, we looked at threshold where the sensitivity and specificity is nearly the same. Sensitivity in the same coverage, specificity means probability of correct prediction. So what happened? Let's say if you increase the cutoff, let's say instead of 0, you take to the 0.5 or 1. What will happen? Only limited number of peptide will be predicted as a positive peptides or the, let's say, antibacterial peptide or whatever. But their probability of correct prediction will be very high. Okay. If we go down 0 or less than 0, even minus 0.5, then what will happen? Too many peptide will be predicted antibacterial peptide, but their probability of correct prediction will be less. So depending on your need, you should keep higher if you want to select only the limited peptides actually. So that's the way. So when we do the research, let's say we publish a paper, we keep sensitivity and specificity nearly the same so that it makes the balance and we provide that threshold. But as a user for you, if you have the limited resources, I will say keep higher threshold actually.
Okay, thank you, sir. Yes, okay.
So now I call upon Pradeep Kaur, coordinator MBSI, to talk something. Yes.
1 minute, sir. 1 minute. 1 minute. No, are asking email ID of Dr. Raghav, sir. Email ID.
Yeah, my email ID is there on my website actually. Anybody is interested, just put the Raghav GPS. Everything is there actually, including my email address and all address.
I will share. I will share with the MBSI.
Yeah.
I will share.
No problem. No problem.
Now, Pradeep, please.
Thank you, sir. Very warm greetings to all. Dr.
Okay.
Arvind Deshmukh, sir, President, Microbiology Society, India. Dr. Swaranjit, sir, President, Microbiology Society, Chandigarh Unit. All the distinguished participants, ladies and gentlemen, and very good morning to all of you. Though I have to be present at the start of the meeting to introduce our worthy guest to you, but due to some bad internet problem, I couldn't be there. So apologies for the inconvenience. So I'm feeling more than delighted and honored to be here to introduce our worthy guest, the distinguished guest we had today, Dr. Raghaya. So I would like to introduce about him, Dr. Gajendra PS Raghaya. He is a professor in the Department of Computational Biology at Indraprastha Institute of Information Technology, New Delhi. He received his MTech from IIIT New Delhi in the year 1986 and PhD from IMTech Chandigarh in 1996. He worked as a postdoctoral fellow at Oxford University from 1996 to 98. He worked as a bioinformatics specialist at UAMS USA and visiting professor at Post-Tech South Korea. He worked around for 30 years at different scientific positions in different fields of the bioinformatics at CSIR Institute of Microbial Technology, Chandigarh. Dr. Raghaya's group has published more than 350 research papers in reputed journals and developed more than 300 in silico products, including various types of the web servers, databases, and software packages, now which is considered to be the highest contribution by any single group in the world. He's a strong supporter of open source software, web servers. All these services developed at his group are free for the academic uses. That is something Dr. Raghaya has discussed during his presentation. So these web services are heavily used worldwide, heavily cited. He has, to his credit, more than 150,000 hits per day. He's highly cited researcher with a 21,000 citation and an H index of 75. He's also recipient of certain major awards, including Shanti School Bhatnagar Award, given to him by CSIR in the year 2008, National Bioscience Award 2006 by DBT, and JC Bose National Fellowship by DST in 210 to 220. So this is about a brief introduction of Dr. Gajendra PS Raghaya. So, sir, it was my gratitude to express my heartfelt thanks to you for accepting our invitation for this webinar. The webinar was quite interesting. It was quite informative. We have gained a great knowledge about the different types of the web servers that you have developed. And the most remarkable thing is that they are freely available, and the researchers and the academicians, faculties, and the students, they can have access to all these web servers. So at the end, I also like to express my gratitude to all the participants of the webinar for their presence and their contribution to make this webinar a success. My heartfelt gratitude and thanks to Dr. Arvind Deshmukh sir for always aspiring us to do more in the field of the microbiology. And last but not the least, Dr. Swaranjit sir for his untiring efforts and for making this, supporting us and making this seminar a success. So thank you once again. Thanks everyone for this wonderful session. And thank you once again, Dr. Gajendra PS Raghaya, for this wonderful session and the talk that you have discussed with us. Thank you once.
Thank you very much. Thank you, everybody.
Thank you, Pradeep, also for the
One
wonderful
minute,
vote of
sir.
thanks.
Please, 1 minute, please. Hello.
Yes,
Hello.
yes.
1 minute, please.
Yes,
in
sir.
the chat box, Dr. Manish Kumar from Madhya Pradesh, he has written one question.
Sure.
So he is again repeatedly telling that please answer my question in the chat box. So I will read it. Okay.
Yeah.
I will read the question, his question. He is asking that, can we go for wet lab experiment based validation for such peptides? What problem may occur in validation?
Sure. I will tell you a very interesting example, actually, that will clear it. What happened in MTech, sometimes I have the colleagues who all are biologists, actually. They challenge me that you are well-known bioinformaticians and everybody using your tools for the different validations and everything. Can you bring a single molecule in real life? So what I did, actually, I collected all the cell-penetrating peptides, develop a prediction method, cell-penetrating prediction method, then take the whole sysprot. Sysprot is the database which contains the large number of proteins, actually, and find out the top best peptides according to the prediction, which can work as a cell-penetrating peptides. Then we synthesize them and we check in real life. All 10 peptides were cell-penetrating peptide, and our best peptide was better than any peptide in the world, actually, including TAT and penetrating, actually. Much better than them, actually. We demonstrated, we published it, and even we are able to deliver the drugs through these peptides, actually. We are able to handle the drug-resistant bacteria through this peptide, attaching the drugs with this peptide, actually. So idea is, if you use the theoretical and experimental knowledge together, then you can do the great jobs. So yes, answer is yes for that.
Good, very nice. So I think we can conclude the session today. Dr. Deshmukh?
Yes, sir. Yes, sir.
Would you like to say something?
You. No, thank you again. Many thanks to sir for best illustration. And this is available on YouTube. I will circulate YouTube link to all, to also sir, to you. Thank you. Thank you very much.
Thank you very much. And I thank Raghav for the wonderful talk. And I also thank Pradeep Kaur for giving us a wonderful vote of thanks. Thank you.
Okay, thank you.